Thank you, Dr. Fakhruddin, and thanks to the organizers to give me this chance. I am not going to teach you breeding. You are all well versed with it. My training in the last 25 years has been looking at questions of why not or why, rather than then we have people who actually deliver those questions into reality. So I have Dr. Ramesh Bhatt is a co-author. He is at the University of Agriculture Science, Dharwad, especially because I was adding few things about genomics. I am a conventional plant breeder. I didn't want to make mistakes. You won't see any big graphs, etc., but I wanted the language to be right. Does it work like this? भाई ये नहीं चल रहा है. Oh, sorry. Okay. So what I want to tell you is globally $650 to $700 million worth of business. China leads in green and India leads in red production. But India yields 50% of red and 38% of green versus China. So that is the gap. Why? That is the question that I am asking. Although there is $1.5 billion worth of exports from India. It's one of the highest out of our export crops from India. So, and the diversity of markets is kind of given in that picture. And don't worry about there are so many segmentations that today are actually being addressed by the private sector. They all have hybrids for each of these segments. And it's totally about 418, some people say 500 crore worth of annual business. And I think 90% today are hybrids in the market that farmers are growing. So these are the 11 known market segments. But you already heard there are 16 GI tags already granted to the Indian chilies, and there are 2 more pending, one from Rajasthan and one from Kashmir. So we need to address that. So quickly, I have basically 6 ideas that I want to share with you. My first thing is, this is not working. So to me, what is happening is our best farmers take the same yield as the Chinese or the Americans are taking, but our averages are, as I showed, 50% less than China, and for both red and short, we are short. So our problem is, we have to help farmers realize genetic potential, improve our harvest crop quality, new germplasm and trait-linked markers for stress tolerance, breed for cuisine-specific traits, red segment genotypes, mechanical harvest. You may not have heard of that. I want us to move towards that. And lastly, the collaborative platform for genomics, and you heard those presentations today, but we are not doing that in our country. So, out of those 6, first, fixing the knowledge gap. As I said, our farmers are not able to realize the genetic potential. There needs to be our cultivation guides are very minimal. We need to make them user-friendly, and we need to make them state-wise, in their own language, in their working against their problems. It is not happening, so we need to make it user-friendly. We need a finishing school, because I think one of our biggest problems is we don't have extension on the ground. That is one of the biggest mistakes that we have. The farmer doesn't have the access to better knowledge, so we need knowledge transfer. And I am proposing 3-tier system of plant doctors, analogous to human health care. We have general practitioners, we have specialists, and we have super specialists for us as humans. I think plants are also live, and we need that care for them. So we need general practitioners for plants. We need specialists to focus on crop protection, soil health, water management, and super specialists addressing complex issues, climate resilience, precision agriculture. Compulsory rural service. China had the same problem. China was the same as us in the 70s, 80s. Today, they are 3 times ours. And what they did 10 years, 15 years back, they created a it's a platform where all the young graduates from this agriculture all over the country, 2 years rural service was made, but they made them ready to be able to transfer the knowledge. So we need to create that ecosystem. And my last and the most important thing is, if we have IAS, we have ARS, I want Indian Extension Service to happen. Please give, as a recommendation from us, we need an Indian Extension Service, because today extension persons are taken as third-class citizens of agriculture. Sorry to say that, I hope you agree with that. They are not given the same respect as a breeder would get or as another scientist. They are equally good scientists. They need to transfer the knowledge. So that's my first point. Second point, we've seen all the enhanced flavors that we have. We have 16 GI tags, more are pending. Each has a flavor that is linked. You can find, you saw with both Derek and Dr. Kang, that they have found lots of sequences that are associated with these flavors or colors, etc. So what we need to do is, reference genome sequencing has to be done. Why should only ABRDC do it? Why aren't we doing it? Why genetic basis of those key traits? We saw there is polygenic, but really how do we apply it actually in the field? Transfer traits to best agronomic lines and strong PPP. That is my last slide also, but please, you saw Derek's slide. They have done 36 places in all over Asia. Same set of lines went for testing, and that is how they were able to get actual data from the field. That's why they were able to find the association, trait associations with their genomic data. So that is not happening. So we need enhancing the flavor and color traits with all the agronomic traits. And for that, the right breeding, as Dr. Mohan Rao was saying, 400, we need 10,000 plants in the F2. We need at least 3,000 plants in BC1 F2 if we are doing a wild enough cross. Then only we will get the right plant to be selected. Third one, this is the tomato thing. Chilli leaf curl virus is today the biggest problem in chilies. This is an experiment in tomato. We were part of that. We did it under a consortium with ABRDC, which is WVC now. You can see the, you can see the spread of the testing they did with the private sector, and that is the result. All I wanted to focus on, the gene combinations of TY2, TY5, or TY2, TY3, TY5 had a more broader spectrum resistance than genes alone. So the time when we came with TY1 only was okay. Today you need multiple genes. Okay. Today you need multiple genes to be able to do that. And I think already Kang and Derek, they also kind of made. So multi-location, multi-year industry, we need to develop this kind of thing in India. But after all this, there is no one bullet. We released Abhinav in 2003 with TY1 heterozygote. But today, we need 2 genes minimum. We may need even more to get a broader, but still, it is not the final bullet. So we need to, so how do we now get the chili leaf curl virus? So we need most significant disease. So National Collaborative Initiative, urgent needed ICR, SAU, seed sector. Please come together, screen germplasm for this, hotspots and controlled inoculations, characterize virus diversity, white flights, weed host ranges. See, each one I can talk for half an hour, but I'm just quickly combine resistance gene pyramiding with genomic mapping to identify trait-linked QTLs, evaluate genotypes showing both white fly tolerance as well as virus, because then only you will get this synergistic model of resistance. Once done, the consortium, public sector should not, they should transfer the technology to the private sector, let them develop their own hybrids in their own different genotypes. You don't need to become a seed company. I've been saying that for 10 years, I'm a bad guy, but you don't need to be a seed company. You can be a seed company in the open pollinated varieties, where private sector will not do because of the IP protection not being there, as I said in the morning. Lastly, 2 more, cuisine-specific trait breeding. I'll shorten it in the sense, we have I don't know, 30, 40 different cuisines made with peppers, right? Different flavors, different colors, different way. We need to add the agronomic traits in those type so that there are 16 GI marketing commercial hybrids for all the GIs in our country with all the agronomic traits we need. So it will not happen over a day. It needs collaboration to be able to give them the right agronomic background with the flavors that they have. And we can also transfer the flavors to that are not having the flavor today. So this needs discussion and developing the topic. Last but one, mechanical harvesting. I have a story here. I was in USA in 1982. This one, Jack Hanna was the breeder. Lori was the engineer, and Black Welder was the tractor developer. They were laughing at this guy when he said, I will develop a mechanically harvesting tomato. Cutting the long story short, 1982 it was already happening. The 500 acres per farmer, he was doing mechanical harvesting. 2 genes, self-pruning and jointless gene, and of course the processing traits like viscosity, color, etc. were able to make $1 billion industry. This is my kind of a field assistant. This we have in tomato. My PhD student did that. We have the genotypes, but it is not working with the industry to take it there. So we need, but see that China. I don't want to speak, the picture speaks. This is China doing mechanical harvesting in Xinjiang area, northwest, because labor, even today, 700 rupees a day is the labor cost. We cannot be depending on labor alone. Red chilies don't have to be harvested 20 times. Red chilies can be harvested once. And we have the genotypes here. These are photos at the bottom that are in India. The genotypes are there. We need to develop uniform flowering and ripening, easy fruit detachment. Dr. Prabhudeva is going to talk maybe of destalking gene, determinate plant, once over picking. You need to use also ethereal, 250 ppm. Once about 20-30% fruits are ripe, you spray, you will get all the fruits at one time. So why do you need to pick a red type 5 or 6 times or 20 times? So genotypes, so develop appropriate. The second point is either this has to happen through FPOs so that a larger area can be harvested by existing mechanical harvesters. But we are smallholder farmers, 1 hectare people. So please, agricultural engineers need to come up with a mini tractor with a mini harvester so that that can also happen. So it is time for discussion. And last slide is, sorry. ये कैसे जाएगा भाई? Yeah, this one. क्या हुआ? Last. Yeah. So no. Last one. Then you go down. Last. Anyway, the last slide is about us doing the right genomics experiments and using all the modern breeding tools. You have had a plethora of, all this was already mentioned by the earlier 2 breeders, and Dr. Mohan Rao also did something like that in another scale. We need to strengthen this work under a PPP mode. I call it pre-compete research. Pre-compete research means it doesn't have to go to commercial at that stage. Once you understand, develop the marker, discovery, then give it to the breeders and let them develop the hybrids. I think these are the 6 from my idea, but please give your thoughts to it. Write us in the small ones, the bowl is missing. Please send us by email. We need to develop this as a policy document that we can give to the government and wherever Karnataka government can also think of. Thank you very much. That's my...
Thank you, sir, for a very thought-provoking, very novel talk. Thank you very much. Now, I call upon participants to have, if you have any questions. Quick few questions. Yeah, there. Mike, please. Oh, very nice, very nice. It was very novel, sir. I have a couple of questions to you. I'll ask later.
Sir, thanks for nice presentation. This vector component is totally neglected with respect to virus. This sir, vector component. What you mentioned the biotypes, vector component is old system as neglected, sir. Then what is your
Viral
use?
vector.
Vector, especially now there is.
White flash. Yeah, you know, today there are reported papers on whitefly resistance in the germplasm. So we need, if you have whitefly resistance as well as the genes, you will have a higher level of more broad spectrum resistance is my feeling. You are more practical, but I feel there has to be some synergistic effect brought into the system. Maybe Dr. Madhavi can add to that, because I think it is a good, because if you have only whitefly resistance, that doesn't give you resistance to virus. You need for both. So that's my idea.
Out here. Yes, sir, please.
I beg to differ, because vector resistance often does not translate to virus resistance.
I already said
Very
that.
often.
I already said
So, let us not be under a futile chase, because in the entire lifetime of plants, one snapshot of vector infection is enough to, if the plant is susceptible, it will become susceptible.
Yeah.
So, let us not try to build human beings resistant to mosquitoes. Let us find answers for malaria. The question that I am trying to tell you in a very different way is, the other point is, Dr. Nijagun said today morning about the very, very elite segment of chili oleoresin extracted through seeds and oils. And I think in the overall chili improvement program, since that is at the top segment, which translates to extremely high money, we are one of the largest producers of the red chilies. I think now this is the time for the India and the entire group to come out with how best we can draw a map so that we really harvest the fruits of the segment, which is going to be most profitable at the end of the day.
Yeah, my last slide was about that. Through the genomics field, because otherwise it will take too long. You can't transfer it by conventional breeding alone. You need a combination. Okay. Some more? Yeah.
Question, sir.
Yes.
Okay, sorry. Sir, my request to all the breeders is with two different aspects, sir. Especially in vegetable crops, including chili. Nobody talks about lines with more nutrient efficient types. We are all talking about yield and quality. But I want low input responsive hybrids. Under normal or ordinary management should outperform as expected. This is most important thing, sir. Second question is with respect to chili. This year, we have lost nearly 11,200 hectares of chili crop in Karnataka alone due to heavy rains and very high soil moisture. Do we have, as breeders or as agronomists, any solutions for these two situations? Thank you, sir.
Number one, you can't fight with nature. You can't get all the genes together in one go. People have been working for 30 years, 40 years, because climate is also changing. So to get a chili that will not get even one disease, you need to also see that it grows under anaerobic conditions, because the water is totally, full of water. So you need a huge, screening to find where those genotypes are. So there are 14,800 accessions that AVI receives. Where have we done the screening? So we have to actually go to the field and do those screenings. Once you have a method that shows problems, then you can permit those genes using the technology that we have. What about the nutrient? There is nutrient use efficiency, diversity available in solanaceous crops. Tomato, we have proved it in section 22. Prabhakar and me have a paper that time. But you have to work on that. At the moment, the hybrid is growing at NPK that you are giving to the varieties. It will give 30% more heterosis. if I am right. Dr. Madhavi, am I right?
Hello, good evening. As of now, just like in other crops, there is no answer for really how to come to the excess rain. Even chili, if the anaerobic condition is created for more than 40 hours in the rhizosphere, crop wilts. AVRDC is doing attempt, I don't know how is the progress in World Vegetable Center now. Brinjal as a root stock for chili, it is feasible. And it is more tolerant than chili for waterlogging, they have found. They have brought out in tomato. IHR has also found our own brinjal hybrids as root stocks for tomato. But chili, this work is not done. If somebody will be presenting from Vienna. It's one of the leading, this one are root stock and the grafting technology in chili. Till genetically it is brought out, it is just like combination of rhizosphere of a crop of mesophytes and hydrophytes in one crop. It is not that easy, I know that. So till that, there are some agronomic manipulations feasible. Tomorrow in my talk, I will be presenting on that on a very small one of the points. Thank you.
Thank you. Dr. Madhavi wants to add something.
Sir, we have tried actually, chili and brinjal are not compatible, graft compatible. That is one thing. They are not graft compatible, like tomato and brinjal. And also, right now, what we can do, proper drainage, and fertilizers, if we can incorporate, that will solve our problem right now. Because breeding for that will take longer time for waterlogging conditions.
See, C335 is an oldest phytophthora resistant genotype and very successfully.
No, we
in
have
Korea,
developed
we had transferred it.
two hybrids at IHR. They are performing very well.
Yeah,
Tried.
but grafting, as far as I know, not as successful in chilies as it is in tomato. I published the brinjal and tomato for bacterial wilt in 78 in Current Science. So we know it is commercially exploited in Southeast Asia, Japan, China. They sell grafted seedlings. Vietnam, they sell grafted seedlings on bacterial wilt and fusarium wilt resistant rootstocks. Please, last one.
In fact, in my presentation, we missed it. Rootstock breeding is in chapter today.
I request
Now we have
Dr.
many
Krishna to
rootstock
load the
breeding
slides.
material already now we are releasing in the market. We have WR 1, 2, 3, 9 breeds which are giving in coastal belt of Maharashtra. They go for grafting, chilies. The rootstock for that is very different. We have material for that, we have hybrids for that also now, which can sustain even a high moisture status. This is one. Second is early maturing hybrids are excellent today, which can be climate resistant. Because today, every day, the expenses by the farmer, when the crop attains 80 to 90 days, the expenses go up like every day 1,000, 2,000 rupees expense to manage the crop, the pest and diseases. So if you can introduce in earliness, we have introduced, this is one of the very important characters to introduce in all the F1, but there are 40, 45 days early to any hybrid today in the market. So these two aspects, in fact, rootstock breeding is a very challenging area, and we have already 3 hybrids released, and many more are in pipeline today. So that's really coming up well, even in India. Thank you.
Thank you. Thank you, sir. Thank you very much. Sir, I have one more last question to you. You mentioned about finishing school. How do you think that we should structure it?
We had given
Meanwhile, I request Katta Krishna to come up and load his slides.
from ATPBR, we had given many times a suggestion, and we would do that from AIH also, that when your graduates are about to, there should be a 1 month or a 2 month kind of an exposure to give them practical crop-wise idea on how to face these, what to do and how. But that needs to a collaboration again between those who are in the field and also in the universities. Find a method by which we can make this finishing school happen. Thank you.